The GENIE study was designed to develop biomarkers to help predict which patients with breast cancer that are receiving one of the most common chemotherapies are at risk of developing peripheral neuropathy. So peripheral neuropathy is something that happens sometimes with taxane chemotherapies. It's pretty common. It happens about 70% of patients will experience it at some point during their treatment, but about 20% of patients will have moderate to severe symptoms that present as either like tingling or numbness in the fingers or hands or feet, or sometimes it feels more like a burning sensation. It can be pretty debilitating. And in a subset of patients, those symptoms can persist for months or even years after the chemotherapy has stopped. And so it can be a very debilitating thing for patients. And it's one of the most common reasons that patients have to discontinue this otherwise pretty effective treatment. So what we want to do is to identify prior to them receiving the treatment, which patients are at risk, so that therefore they could have decisions can be made about their care, whether or not this is the best treatment for them, or maybe they need a modified dose to mitigate that risk. And so what we're doing is we're doing a multiomic approach. We're doing 6 different omics technologies to get a really comprehensive characterization of the molecular profile of the patient, both prior to them receiving it, as well as during and after their treatment as well, to develop and use machine learning to identify the best combination of those molecular features that can predict which patients are at risk.